Hi everyone, back again for another lecture. Today we're going to be talking about inheritance. Now, just as a reminder, I've kind of filmed these out of order a little bit, just kind of what fit into how I was doing it. Oh, of course it's going to start acting up now, come on. That's not what I'm doing. There we go. So hopefully you're following along in your PowerPoint, whatever way you feel like is appropriate or best for you. So we're talking about inheritance, so I'll probably pull up the whiteboard at some point, today. So we're going to talk about inheritance. We'll jump right into Gregor Mendel, first slide. So I'm assuming that most of you probably, in like high school or something, probably got a little discussion on Gregor Mendel. So we sometimes kind of refer to him as like the father of modern genetics. He was the first really to take the steps to understand this in like a very scientific process way of like, what are the rules for how things are passed down? A lot of people thought before, whether it was plants or animals, was that traits were kind of blended, because sometimes traits do kind of appear to be blended. Like sometimes, some of them do. Like a good example is height. Like I'm 5'10", my mom is 5'4", my dad is 6'4", so I'm literally like right in the middle, right? And sometimes in plants, colors can look blended, right? But we know genetics are way more complex than that. But at the time, they didn't know any of that. They didn't know whether it was complex or simple. They didn't even know what these words were. They didn't know what a chromosome was. They didn't know any of that. Interestingly, right at the same time that Gregor Mendel is doing these very famous and very vital experiments, Darwin and Wallace were presenting their ideas to their colleagues about natural selection. And really, the one thing that most of those other people that, in the scientific community didn't kind of had questions about for Darwin was this idea of how things are inherited, because at the time they thought traits just were kind of blended. And so they were like, okay, Darwin, your idea kind of makes sense, but you're saying that certain traits are going to be more beneficial in certain environments, so they'll pass it down to like their offspring, but how can that really happen if traits are just blending away? They didn't understand like they're actually not being blended, right? There's a whole pattern of how this works, and dominant and recessive, all the stuff that Gregor Mendel was figuring out around this same time. It wasn't, unfortunately, until a few decades later that people actually combined this info from both of these individuals and was like, oh, this all goes together, right? Like now we live in a world where you can just go into Google Scholar and see what the most recent research is on like any given topic, and see, oh, these two fields, working on something that's kind of similar. It's not the same as it was back then, right? Okay, next slide. A little bit more on some of these terms. So this is we've gotten this in one of the other PowerPoints, right? One of the other lectures, but just to kind of go over this. So remember the terms like gene and chromosome, right? So it's kind of showing you a visual, but I'll just draw it again. So remember that our chromosomes are paired, right? And if just to reiterate, we have, cells in our body, and in those cells we have chromosomes, and chromosomes are made up of all the DNA, right? It's basically compacted and uncompacted to make these different shapes. Every different species will have a certain number of chromosomes. That's like, the number for their species. For humans, it happens to be 23. They're all in pairs. Sometimes they're actually attached, sometimes they're not, but they're still in pairs, right? So if I was like, okay, let me see if I can draw this with my left hand so I don't have to move around as much as before. So let's say we're like, here is, chromosome 11. It doesn't matter, right? It's a loppy chromosome. And here's its, pair. There's also another chromosome, right? We would say, oh, here is a gene. A literal, like, physical location on that chromosome. That's a package of information that is coding for hair color, eye color, nose shape, whatever it is, right? And on the pair, it's going to be the same gene, right? It's going to be coding for hair color or eye color. It's the same thing. Now, remember that allele would represent whether that is the same or different. So even though both of those, the gene, the section of that chromosome is coding for the same trait, hair color, eye color, nose shape, whatever, it can be different or it could be the same. This could be saying, round nose. That person would have a round nose. This could say round nose, pointy nose. And then it would come down to what are those rules of dominant and recessive to what we would actually see on that person, the actual phenotype on that person, right? And that's getting into what this PowerPoint's saying too at the two bottom bullet points of those terms. Genotype being what's going on genetically, and then phenotype being what you're actually seeing on that individual. So the phenotype is the actual physical trait. So like I could have a phenotype, for blue eyes, but genotypically on my genotype, the alleles could be like blue eyes, brown eyes, and maybe one's being, one is dominant, one's recessive. So keep those ideas in mind. We're going to come back to those. And Gregor Mendel was the first to kind of figure this out. So you can see on the next slide it says alleles. So just getting into that a little bit more, right? I'll give you guys a couple of examples, kind of what I already did. You can see a visual of like a pair of chromosomes and a gene, and like blue and brown. It could be like brown eye coloring or blue eyes, right? So the allele is just the alternative form of the gene, right? The gene is basically just that section of that coding section. The allele is really what is that information telling us. So now moving on to where it says alleles continued, alleles. Now, once again, these rules of whether it's going to be dominant or recessive, you know, like I said, Gregor Mendel, when he's doing this research, is starting to figure out, wow, there are actually rules to how this is all working. When certain traits are passed, how they're passed, do they skip a generation? Like, he's figuring out all these rules and stuff, right? And so, like I said, continuing on this slide, you can just go through those bullet points, and it says, like, you know, dominance is the ability for one allele to mask or hide another. So basically what I was saying, phenotype, you might just see, oh, they have blue eyes. But genotypically, in their actual genes, you know, it could be, one allele could be for blue eyes, but what the other allele could be for green eyes or something like that. Actually, like eye color genetically is actually much more complex than that, but this is fine to kind of give this as an example. So you can see here, remembering that they're always in pairs. These genes are always in pairs. These alleles are always in pairs. There are sometimes ways that we can kind of use letters to represent these pairs, right? Now remember, we are always talking about dominant and recessive, whether an allele is dominant or whether it is recessive, right? So two options. Now, we use letters to represent this. Now, you can use any letter from the alphabet that you want, as long as the letters, uppercase and lowercase, look different. Like, I can't do it left-handed. Don't use C. They look exactly the same, right? Don't use S, because you have to make it really obvious that it's something different, right? You want to use a letter where the uppercase, even if they're the same size, that the uppercase and lowercase look different. So I like to use A, because even if I drew them the same size, that you know which one's the uppercase, you know which one's the lowercase, right? You can use R. They look the same. I mean, they look different, right? So it's really up to you. I don't care what letter, as long as, like I said, the uppercase and the lowercase. Oops. The uppercase and lowercase look different from each other as we go through these. So I just want to point out, though, since we're talking about, you know, dominant and recessive, there's only 3 ways you can combine those together. Oops, it's not dry. There's only 3 ways you can combine. Either you have 2 dominant, right? You have 1 dominant and 1 recessive, or you have 2 recessive. If our only options when talking about these scenarios of paired genes, you know, here they are in pairs, and we're only dealing with dominant and recessive, there's only 3 ways we can combine all those together, right? Either two big A's, 2 dominant alleles, 1 dominant allele and 1 recessive allele, or 1 big A, 1 little a, or 2 recessive alleles, 2 little a's. This is an important concept, so hopefully if you didn't understand it, like, rewind it and go back. This is important to know. That when we're dealing with dominant and recessive, those 2 things, there's only 3 combos that you can get from those 2 things, right? Okay. So the slide is kind of telling you that, right, that last little part. So there are 3 allele states, and these are the 3. We call this homozygous. I don't really need to write this out. It's on the thing. I won't even write that out. You guys can see it on the PowerPoint. Homozygous dominant. So homo meaning the same, 2 dominant, heterozygous, and then homozygous recessive. So homo meaning the same, and then 2 recessive alleles, right? I'm sorry, I shouldn't have even started to write that because you have it on the PowerPoint. Try to make the writing on this not as much to make it easier for you guys, because you're like, what is she writing? And she's wiggling the board, whatever. Okay, so moving on to the next slide. So here we have an example of a few what we call Mendelian traits. So Mendelian traits are when they are actually really simple genetically. It's like 1 gene, 1 trait. It's not very complex, like genetically. Some stuff genetically is really complex, right? You're pulling info from different genes, you know, like a bunch of stuff, right? So sometimes they are simple Mendelian traits. So there I have a list of a few. So one is, don't think about it right now, just do this, do that. Now, if you're looking at your hands, look down, you're going to see one of the thumbs is on top. Is it the left thumb or is it the right thumb? You actually have a gene that codes for how you do this. Now, if you do it all in the opposite way, like not just the thumb, but like do all your fingers the opposite way, it feels weird, right? It doesn't feel normal because you actually have a gene that codes for that just instinctual behavior to do it in a very particular way. Most of the time people have, it's the left thumb on top. But yes, you actually have a gene that codes for that. So that's like one gene, one trait. Here are a few more examples you'll see on the PowerPoint slide. So earlobe attachment, whether someone has attached earlobes or unattached earlobes. So I have unattached earlobes. I have a lobe that kind of hangs down. Some people, they have a lobe that just kind of goes into the side of the face. So that's like an attached earlobe. Darwin's tubercle, you'll see on the PowerPoint slide. So that's a little nodule. Most people have this little nodule like at the top of their ear. It doesn't really have a function, just, you know, some people have it, some people don't. You have one gene that codes for whether you have it or not. Cleft chin is one. One gene, one trait. You either have a cleft chin or you don't. The widow's peak hairline, so that's when like your hairline, oh God, the hairline kind of comes to like a point right here. Sometimes I'll draw mine on. I think it looks really cute. When I don't have my bangs on, it's really cute. So you can see Evangeline Lilly in the middle has that nice, you know, point to her hairline. So those are all examples of when there's just, it's simple genetically. There's like one gene, one trait, you know, simple. Moving on to the next slide. So now we can kind of take some of these ideas and kind of work with them a little bit. So let's pull out the whiteboard again. So now we have an example of flower coloring. Oh, maybe I don't need, oh yeah, I do this. Okay, so here it's saying, this is just an example. We're looking at flowers and we're looking at colors. It's telling us, for this scenario, there are two colors, purple and white. Now white is telling us, I'm sorry, purple is the dominant of the two. So the dominant one is purple, and I'm literally rewriting what it says on the PowerPoint slide, but we've got, you know, purple is dominant, and then white is recessive, right? White is recessive. And it asks one question. What are the flower colors for the following genotypes? And then it lists basically the three combos. So if we're given these three different genotypes, that's what's going on genetically. What are we actually going to see on the phenotype of that flower? What's actually going to be on the outside, what you're going to visually see when you look at that flower? This is what's going on genetically that you can't always see, right? What are you actually seeing on that individual, like on their phenotype, right? Given that those are the rules right there. So for a flower, I'm trying to keep the glare out. For a flower that's homozygous dominant, big A, or, you know, dominant allele, we know the dominant state is purple. So basically what their genes say is purple and purple. So, you know, simple, right? It's going to be purple. That's all they have. It's super simple. For the individual flower who is heterozygous, they have one allele that says purple and the other allele says white. But remember, according to the rules of dominant and recessive, the dominant will mask the recessive. So even though genetically they have that allele for white, it's going to be masked phenotypically. So what you're going to see on that individual flower is still going to be purple. So that's a really important concept. And then the third option, that individual who's homozygous recessive, all they have is one allele that says white, the other allele says white. So obviously that's what they're going to be, right? It's white, the white coloring. Now you could say also in addition, there's no dominant allele masking the white. That's fine too, right? So we're going to use this concept for a lot of these different exercises. Hopefully this makes sense to you. Like I said, it's not complex, but I know sometimes with all of the little examples it can get a little tricky. So hopefully, like I said, you can always rewind and make sure you understand this part of dominant versus recessive, you know, masking, hiding something, genotype versus phenotype, that whole thing. So moving on to the next slide, Punnett squares. We're going to do a couple of these together. So Punnett squares, as you can see on the image on the PowerPoint, it's giving you an example using the letter A, which is what I use. So it's showing you like on either side of the square you would put like the male or the female, or like the mom and the dad, and then you would fill in this little square with the possibilities for their offspring, right? And we're going to keep with those concepts of, you know, dominant and recessive as we do this. And so basically I'm gonna give you some information, or the example will give you some information, and then you will fill in the square and then answer some follow-up questions. So There will very likely be a question like this on the exam, and we will do, I think I've got either one or two, oh, looks like I got quite a few examples on this PowerPoint. So let me see how many. Okay, we'll do the first one together. So Punnett square example 1. So you can see it's got, oh, I blanked on her name now. Allison Hannigan. So she's got that widow's peak hairline, right? The widow's peak. So that's the trait we're looking at, and it really doesn't matter. We could be looking at any trait. This just kind of gives us a nice visual to kind of follow. So we're looking at this particular trait. So we're going to be doing these Punnett squares together. So here we have the widow's peak hairline is a dominant trait. Keep that in mind. We're going to come back to that. And it's telling us there are two individuals, Matt and Laura. Matt is homozygous dominant, and Laura is heterozygous. So using, the Punnett square, we're going to draw the square. Oops, I just put the pin that, of course, doesn't work. Oh, just made a mess. Okay, let me just draw. Okay, so here I have the square. I'm going to make, it have 4 squares, kind of like a window. And it doesn't really matter if you put the male or the female on the top or the bottom, right? We'll put Laura up here, and we'll put Matt over here. And it's already telling us Matt is homozygous dominant. Homozygous dominant. Homo meaning the same, dominant. So he's got 2 dominant alleles. So we're going to put one big A here and one big A here, kind of in those next to those 2 rows. Sorry, the sun is, setting, and I'm getting a little, it's like golden hour right now, but if I have to, I'll go up and close the blinds. And then Laura is heterozygous, so she's got 1 dominant and 1 recessive. And basically what you're going to do is you're going to pull these letters over and down, and you're going to fill this in. So each of these other 4 squares will have 2 letters in them. So you can see here I pulled this one over, this one over, this one down, and this one down. Hopefully that makes sense to everyone. And so now, like I said, you're left with 2 letters in each of those individual squares. So this is basically showing the possibilities for the offspring of Laura and Matt, at least for this one particular trait. And then there are a couple of follow-up questions. The first is, what percentage will be heterozygous? Heterozygous is 1 dominant, 1 recessive. Hetero looks like 2 will be out of the 4, so we could say, 50%. The next question says, what percentage will be homozygous recessive? Homo meaning the same, homo recessive, that would be 2 recessive alleles. That's a 0, right? For these 2 individuals, there's a 0% chance that would happen for them. And then the last question, which will show the trait? So show it means like phenotypically. Who's going to show that trait phenotypically? It looks like everyone will, right? Because it's already told us that the widow's peak is a dominant trait. So whether you're homozygous dominant or heterozygous, you will show that trait. So if you're homozygous dominant, obviously you're going to show the trait because you just have a trait that says, widow's peak. So you're going to obviously have the widow's peak. But for those 2 who are heterozygous, even though they have another allele, the recessive allele that says no widow's peak, the one allele that says widow's peak is masking or hiding that one. So phenotypically, on the outside, you will still see just that dominant allele. You will see. So we could say 100% of these individuals will show a widow's peak hairline. Okay, let's do another one together. Use a whole bottle of cleaner. The next one, Punnett square example 2. So now we're looking at hair types, so straight hair versus curly hair. Let's see, so we've got straight hair is recessive. Let's do straight hair. And then curly hair is dominant. Okay. We've got 2 individuals, Dan and Lisa. Let's do that Punnett square. Remember, it doesn't matter where the male or the female go. Let's do Dan and Lisa. And Dan is heterozygous, so 1 big A, 1 little, or 1 dominant, 1 recessive. And then for Lisa, she is homozygous recessive. So homo meaning same, recessive, so 2 recessive alleles. So 2 little a's. And then we're just going to pull those letters down and across, so where we end up with 2 in each of those squares. And then we can answer some of the follow-up questions. There are only 2 for this one. So it says, what percentage will have straight hair? Remember, straight hair is the recessive straight. So for someone to have straight hair, they cannot have a dominant allele masking it in any way. So for those who have straight hair, they have to have 2 recessive alleles. And look, we've got 2 of those. 2 out of the 4, right? So that's 50%. And then it asks, what percentage will have curly hair? So curly hair, as long as they have at least 1 dominant allele, or 2, they're going to have curly hair. And we have 2 out of the 4. So 50% will have curly hair, 50% will have straight hair. Make sense? Okay. So there's a third one. You guys can do that on your own just so we can kind of move along, but it's kind of similar. You're going to set up that Punnett square. This one's for Brian and Amy. And it's going to ask about the cleft chin. So just, actually, a good practice for you guys to do that one on your own. And in class, maybe I'll give you the answer so you know if you did it right or not. We'll do that. Okay, so now, Punnett square example 4. I didn't realize I had so many. Probably just for you guys to practice with. I think I threw a few in last semester for that purpose. Yeah, so there's actually 2. The next one is Kevin and Lyra. And it's talking about the earlobe attachment. And so same thing. So like I said, I've done 2 for you. You guys do the other 2. Maybe I'll do those other 2 in class or something. Just so, like I said, with a few of these topics, I probably would reiterate a few things in class so you guys have the chance to ask questions. So this will be likely one that I will do. So that actually kind of works out for us. Okay. So moving on to the slide that says sex-linked inheritance. So this is just referencing the fact that on all the chromosomes, right? Like in humans, we have 23 pairs of chromosomes. Our 23rd pair are the sex chromosomes, right? If you're a female, you have two X's. If you're a male, you have an X and a Y. It's not as if only on those chromosomes, it's just information about who you are in terms of your sex, development or traits or something. Sometimes there are other traits that just happen to be on the 23rd pair of chromosomes, but because they are obviously linked to either an X or a Y, we call them sex-linked traits. And so sometimes you might see them being more prominent in males or in females depending on that. So if you go to the next slide, you'll see it's a sex-linked example, and there's like 3 circles. Most of us can see, in all 3 of those circles, we can see a number, but not everyone will. And this is usually when I do this one in class, I get at least, especially in the lecture when I have a few more students, I usually get at least one person who's like, I can't see all the numbers. This is a test of whether you're colorblind. And there are like different types of colorblindness, right? But this is something that occurs, on the X chromosome. So if you're a female, both of your X's have to be affected for you to have X-linked colorblindness. But for a male, you only need just your one X to be affected. So it's much more common for males to be colorblind. So think of someone in your life who's colorblind. Maybe you know someone, maybe you are. Almost always it's going to be someone who's male. It can happen in females. It's just much less likely to happen. And then the last slide talks about a couple of Mendel's laws. I put those up there. I really don't reference those much more or anymore really, but that's just kind of giving you a little more detail on what we talked about with Mendel. And that's it. Wow, that's it for that one. So quickly through, only 20-something minutes on that one. Like I said, moving through these quickly so you guys can just, you guys can have them. You can rewind, pause if you need to. In class, I will reiterate a couple of things, but, keep this lecture part short and sweet so we can jump right into those labs and stuff. Okay, see you guys in the next one.